Now let us start our study with the Human Genome Project. Human Genome Project is said as HGP in short form. Okay, now what is Human Genome Project? This project was proposed in 1990 and is considered as an international scientific project whose primary goal was determining the sequence chemical base pair that makes up the DNA. And to identify and map about 20 to 25,000 genes of the human genome. Thus, with the Human Genome Project, it was obvious that the gene, their mapping and their usage, that is usefulness to mankind, could be obtained. In this manner, the sequencing of the segment of DNA, that is the base pairs which make up the DNA and helps in identification of such DNA segments forming genes, was done within the human genome, and so it was named as Human Genome Project. It was proposed and made in 1990. This figure depicts the human genome map that was found or made in 1990. This figure depicts the genome structure of different organisms, that is humans, their genetic structure and the arrangement of genes within it. In 1990, this project was initiated by the joint efforts of the US Department of Energy and National Institute of Health. That is, these two centers together found and made the Human Genome Project. However, a working draft of the Human Genome Project was officially announced in 2000, where the analysis of the working draft was published in an official manner. Now, in order to prepare the Human Genome Project, there were a few goals that was established in order to set this project. These goals were to prepare the genetic map of all genes, that is, the genes that are present within the human beings, to analyze and make a particular genetic map of it. Second goal was to understand or to store the information in databases and improve the project tools These tools were necessary for the various analysis of the project, that is data analysis, and helps in the transfer, that is the related technologies, to the various sectors. Following it, this information which is stored should be of nucleotide sequences of each and every gene and moreover its expression should also be determined and stored. Apart from this mapping of genes should be considered or should be made under the consideration of social, ethical, and legal complications that is related to the project. And these were the complications which was necessary to study and thereby find proper solutions to them. Thus, these all were the main goals which were set in order to understand and construct a human genome project. However, in April 2003, this HGP sequencing, that is Human Genome Project sequencing or mapping, was completely done. On this basis, it was calculated that human genome contains about 3 billion nucleotide sequences. That is the sequences that are formed due to nucleotide bases. These bases includes adenine, guanine, cytosine, thymine. These are the bases which constitute the DNA. However, on an average, gene consists of 3000 bases. And the total number of genes that is estimated is about 35,000. Which was lower and not proper, because later as it was calculated, it was about 80,000 to 1,40,000. This deciphering of nucleotide sequence and its store is referred to as database. As said earlier, this database information is necessary to store, which occurs in the form of deciphering nucleotide sequence. and therefore it is this database which is necessary to store in order to understand the human genome. On this basis, it was said that the objective of HGP was to understand the genetic map of the human species. In this manner, scientists also obtained the information about the gene or genome mapping about few other organisms like humans. As we all know that the scientific name of humans is Homo sapiens, whose genome size was about 3 billion, or it can be even more than that, and GS means genome size, and estimated number of genes was about 30,000. ES, that is estimated number of genes. Following it, the other organism was fruit fly, whose genome size was about 137 million, and estimated number of genes was 13,000. The scientific name of fruit fly is D. melanogaster. The third organism that was studied was bacterium, with scientific name E. coli. In it, the genome size was about 4.6 million, and the estimated number of genes was 3,200. The other organism that was studied was laboratory mouse, with scientific name M. musculus, whose genome size was of about 2.6 billion, and the estimated number of genes was about 30,000. However, after constructing the Human Genome Project, there were also a few challenges which has to be achieved after. So, the future challenges of Human Genome Project includes to understand the gene regulation, then DNA sequence organization, third, coordination of such a gene expression, then calculating the gene number, their exact location and function. Apart from this, to understand the disease susceptibility, based on the gene sequence variation. Thus, these are all the goals that are set in order to precisely understand about the human genome and its function.